Hopefully you can see molecular genetics. Tell me if you can't.
Yeah, him here. Oh, shut up.
Okay. Molecular genetics. Now the first part of this is just going to be review. I started out with restriction endonucleases. And on this slide, I want to make a couple of points that you need to remember. Now, these are the site-specific endonucleases. And remember that only nucleases among the depolymerases, the major classes of depolymerases, it's only the nucleases that can have site specificity. So you don't find site-specific proteases or lipases or carbohydrases, but the endonucleases can either be specific or non-specific. So there are 5 types of known site-specific endonucleases. They're listed here. The 2 that I want you to remember are type 2 restriction enzymes. These are the ones that their recognition sites are palindromes. It means they read the same forward and backward. They can be from 4 to 8 nucleotides long. And there are 2 things that are of particular use to molecular biologists. The first is, underlined here, the recognition site and the cleavage site are the same. In other words, the enzyme looks for a place in the DNA sequence that it recognizes, and then it cuts it at that same spot. The other important point here, also underlined, is that some of the type 2 enzymes make what are called sticky ends. And these sticky ends are very useful for gene cloning. The other enzyme I want to emphasize here is type 5 in yellow. Cas9, associated with CRISPR, is a type 5 restriction enzyme. And what's important about Cas9 is that it is site-specific, but you choose the site. The investigator chooses the site by synthesizing a guide RNA, which corresponds to the target sequence in the genome. And once that guide RNA binds, Cas9 comes along and makes a cut at that site. So in that way, it is a site-specific endonuclease. The type 2 and type 5 are the ones that are important in molecular manipulation. Now, DNA ligase is here. It's not a restriction enzyme, but it always goes along with restriction enzymes because once you make a recombinant molecule, the gaps in the sequence have to be filled in, and DNA ligase does that.
Professor?
Yeah.
What does it mean to be non-palindromic?
It's non-palindromic, anything that doesn't read the same forward and backward. Let's see. Yeah, here. So if you look to the lower left, it says palindrome in red. So GAATC, if you look at EcoR1, on one strand, it reads GAATC. On the other strand, it reads backwards, GAATC. So any recognition site that doesn't read the same backward and forward would not be palindromic. And you remember I showed you examples on the lower right here of palindromes. Manaplan, Canal, Panama. If you read that backwards, it says the same thing. A man, a plan, a canal, Panama. And then there's another one. So does that answer your question? If it doesn't read the same forward and backward, it's not a palindrome.
Yes.
Okay. Oh, it can be very confusing when you first start working with restriction endonucleases because of their names. They have these seemingly nonsensical names, but they're really systematically named. The first letter in the name of an enzyme is the first letter of the bacterium that produces it. And remember, all restriction enzymes are produced by bacteria. Eukaryotes do not make them. The second two letters, so the first letter is from the first letter of the genus. The second two letters are from the first two letters of the species. So EcoR1, E comes from Escherichia, CO comes from coli, and R1 is the strain of Escherichia coli that enzyme came from. So another strain is EcoR2, another one is EcoR5, and so they're named accordingly. So similarly, Haemophilus influenzae makes a number of restriction enzymes. One of them is HindE3, H from the genus, IN from the species, D3 from the strain. And similarly. So keep this in mind. It's important that you know that. You may be asked about it. And we've already gone over this. I want to make a point about what plasmids are. They're circles of DNA, usually circles. They always come from bacteria. They replicate within the bacterial cell independently of the chromosome. They have functions, usually for toxin production or antibiotic resistance. Some of them have no known functions. And this is what you need to know about plasmids as cloning vectors. So this is an example of a plasmid cloning vector, and it has the bare minimum of what's essential. So the blue here is indicated as ORI, O-R-I. That's the origin of DNA replication. It has to have one of those in order to replicate in bacterial cells. The green, AMP-R, that's a gene for resistance to an antibiotic, in this case, ampicillin. So it's called AMP-R. It has to have an antibiotic resistance gene in order to be selected. If you plate the bacterial cells out, you want only those cells that get this plasmid. And you do that by plating them on a medium containing ampicillin, so that only those with the AMP-R gene can grow. And finally, there needs to be a cloning site. So that could be as little as a single restriction enzyme site, but for purposes of general use, you want what's called a polylinker, or a multiple restriction enzyme site. So you see at the top of this diagram, there are all these sites, EcoR1, SAC1, KPN1, and so on. They're all 6 cutters, they're all palindromes, and they all make sticky ends. So it's important that there is a polylinker there. There has to be at least one cloning site, and it's much more convenient for an investigator if there are multiple cloning sites. Then you have a choice of the enzyme that you'd like to use. Any questions about the essential components of a cloning vector? plasmid cloning vector. I went over this before. Here you see the effect of sticky ends. In this case, the plasmid cloning vector was cut with EcoR1. The foreign DNA that you want to clone was cut with EcoR1. So these are complementary sites in the vector and the DNA. And the ends are sticky. So they anneal together. And then at the end, you have DNA ligase sealing the gaps, and you end up with a chimeric plasmid. And again, we went over this before. Here, this is something that you need to remember. Reverse transcriptase. What is it? Well, going back, you may recall when we talked about the central dogma of molecular biology. In the early days, people thought that there was just a one-way flow of information from DNA to RNA to protein. And in the 1970s, it was discovered that, that it's not a one-way flow. It can be reversed from RNA, single-stranded RNA, back to double-stranded DNA. And that is done by an enzyme called reverse transcriptase. That enzyme is used a huge amount in molecular biology and in cloning. So remember reverse transcriptase, remember what it does. It converts RNA to DNA. Now I'd like to switch topics to polymerase chain reaction, PCR. This is a technology that has truly revolutionized molecular biology. I went over this slide before, and what you need to remember is that here in the upper left-hand corner, what is PCR? It's the synthesis of large quantities of a specific piece of DNA. So the starting material can be a small quantity. It can be as little as a single DNA molecule. And then as you run cycles in this chain reaction, you keep increasing, exponentially increasing the number of target molecules that are being produced. And I went through this before, and you can relook at it. You have a target sequence, and what's critical in PCR is temperature. So first step in PCR is to heat, which causes the DNA strands to separate. And then in the reaction mixture, there are three things. There are primers, specific for the target. There are the bases need to make new DNA, AT, GC, and there's this enzyme Taq polymerase, which I'll mention more in a minute. But first you heat, and then you cool so that the primers can anneal to their target sites, and then you raise the temperature so that DNA Taq polymerase can synthesize new DNA. So now you have two molecules where before you had one. And then you repeat the cycle. You heat, cool, and reheat, and now you have 4. Do cycle 3, you have 8, and if you remember, if you go to cycle 25, you have 33 million copies, starting with 1, and this can be done in a matter of a few minutes or an hour or so. So what's critical here is DNA polymerase, Taq polymerase. It was isolated from a bacterium, Thermus aquaticus, from a hot spring in Yellowstone Park. It's stable. That means it does not denature at temperatures as high as 98 degrees. And that's important because the first step of PCR is high temperature to separate the strands so the primers can bind. So this is one of the few enzymes that is stable at such a high temperature. And it functions optimally at about 70 degrees C. And I won't go through this procedure. I did it once before. Are there any questions about how PCR is performed and why it's so important? You can start with a very little DNA and in a test tube, make millions of copies. You don't need to do it in a living cell. Prior to PCR, the only way you could produce DNA was in a bacterial cell. Went over this before. Now, I wanted to make a point about how PCR came to be, and it centers around a particular scientist who was a very strange person. It illustrates how people with unique personalities, very unusual personalities, can make great contributions to science. So let me just go through the points here. And the person I'm talking about is Kary Mullis. His pictures are on the right. Prior to the 1980s, the only way to get DNA was to isolate it from some organism. And if you wanted to do that fast, you used a bacterium like E. coli because it grows fast in test tubes. A critical point was in 1969, when a man named Thomas Brock isolated this new species of bacterium from a hot spring, and from it was able to isolate this DNA polymerase called Taq. Taq, T from the genus, AQ from Aquaticus, the species, Taq DNA polymerase. And it's able to withstand very high near boiling temperatures without falling apart. Now, in 1979, 10 years later, a biotech company in San Francisco called Cetus hired Kary Mullis for a particular job. Didn't matter what. The point is that Kary Mullis did not do what he was hired for, but rather fiddled around with a lot of other things. And in 1986, he reported at a meeting in Cold Spring Harbor, he reported PCR. And he called it polymerase chain reaction, and he published it. And an interesting anecdote here is that after publishing about his chain reaction, he received a letter of reprimand from one of the governmental atomic energy agencies for publishing a chain reaction without permission. They were thinking chain reaction as in atomic chain reaction rather than molecular. Then, in October of 1993, Mullis was awarded the Nobel Prize in Chemistry. So this is just, 7 years after he first reported this, he got a Nobel Prize for it, which is an indication of what a major impact this development made on the world of science. Another interesting anecdote here, he went to Sweden to give his Nobel speech, and in it, one of the things he said was that he had no joy, no pleasure the night he proved that PCR would actually work, because his girlfriend left him that night. And why did she do it? Well, because he was spending all his time in the lab working on PCR and spent very little time with her. Anyway, a final point here is financial. So out of gratitude to Kary Mullis for developing this technology, CETUS gave him a $10,000 bonus, then turned around and sold the intellectual property for PCR to a pharmaceutical company, Roche, for $330 million. So Mullis did not benefit appreciably, at least not financially, from his work. Okay, I will only talk about what's on the left side of this slide for the sake of time. So PCR is used whenever a large amount of DNA is needed, and the amount of target DNA is small, as little as one molecule. So here are some examples of what PCR is used for. So analysis of clinical specimens for infectious agents. And I want to emphasize here SARS-CoV-2, the coronavirus. PCR is the basis for the standard CDC test for this virus, test that's done millions of times a day. It's good for identification of mutations that cause genetic diseases. In forensics, it's particularly important because any source of biological material, like a blood stain or hair, semen, if there's just the tiniest trace of some biological material that has at least one molecule of target DNA in it, it can be amplified into as much as you want and then used in a court proceeding to prove either the innocence or the guilt of an accused. The next point here, ancient DNA, we're going to talk later about where we came from and how we got to be the way we are. And much of what we know about that has come from ancient bones, mummified tissues, and fossilized material. And again, a bone from a skeleton 10,000 years old can have at least one DNA molecule in it, which can be amplified by PCR to get as much as you want for analysis. And molecular genetics research in all labs, it's used on a daily basis. So the bottom circled in red are two critical points. One, the discovery of Taq polymerase in 1969. And secondly, that CETUS hired this character named Kary Mullis and did not really make him do the job he was hired for, but allowed him to explore. And he came up with PCR. And the question is, and it's a moot question, but if CETUS hadn't done that, and if Mullis hadn't done that, would we have PCR today? And would anybody else have come up with this technology? And if so, when might they have done it? I'd like to just make a quick point about gene therapy. What is it? It's the delivery of nucleic acids into a patient's cells as a drug in order to treat disease. This would be to treat the cause of disease rather than the symptoms. And what you see in the diagram on the right here are two different ways that gene therapy can be done. One is by gene addition. If you have a cell with a chromosome with the red dot, that's a mutation. And if you have a gene in a retroviral vector, remember a retrovirus transforms from RNA to DNA and incorporates it into the host genome. So using such a virus as a vector, you can put in a new copy. A cell that has a defective copy, to that you can add a new copy, and the new copy will function to correct the problem. Another way to do it is to take a good copy of a DNA segment, put it in a mutant cell, and have with the proper nuclease, proper engineered nuclease, have a recombination event where you simply recombine out the mutant gene or region and it leaves a cell with just one good chromosome. The main problem, why aren't all genetic diseases being treated? Some of them have already with this technology, but the main problem is delivery. How do you deliver a gene to a defective cell? And slowly, scientists are figuring out how to do that. And now I'd like to spend some time on genetically modified organisms, GMOs. And the reason I've chosen this for this section is that we live with GMOs all the time. Whether we like it or not, they're there and we're dealing with them. So I want to make a couple of points about GMOs. There are two technologies for making a GMO at the top of this slide. One is with eggs and sperm. This is traditional breeding. It's been practiced by humans since the beginning of agriculture for about 13,000 years, such that now essentially all domesticated plants and animals, everything that we use for either labor or food, is genetically modified by putting sperm on eggs. Example, dogs. All dog breeds have the genome of a wolf. Over 99.9% of the genome of any dog is wolf sequence. So what humans have done is started with wolves and selected for the phenotypes that were desirable, and ended up with the dogs that we see now, which are, you see in the picture here on the right, they're extremely pleomorphic, from tiny little Chihuahua to a Great Dane. They vary tremendously in size, and they're also phenotypically very different from a wolf. Yet they all have a wolf genome. So what counts for that? Differential gene regulation, which we've talked about before, and we will talk about more again. Interestingly, dogs and wolves are, to this day, despite all the breeding and selection, dogs and wolves are still interfertile. So that's one way to make a GMO with eggs and sperm. Another way, available only since the 1970s, is manipulation of DNA, called recombinant DNA technology. So what's the difference in red? The difference between a traditional GMO made with sperm and eggs and a contemporary one is not the type of product, but rather the technology. So you can end up with the same type of product with either recombinant DNA or eggs and sperm. And it's the technology that's different, not the product itself. So I want to, on the lower left here, genetic engineering is precision breeding, whereas traditional breeding produces many unknowns. And that's illustrated in this diagram. So with traditional plant breeding, this is plant breeding, the same would apply to animals. If you have a gene, the gene is a little orange dot here on the yellow chromosome, and you want to get this gene into another plant. And this plant has a green chromosome. So what you do is make a cross between yellow chrome chromosome and green chromosome. And what you end up with is a chimera of components of the yellow chromosome and components of the green chromosome, along with the gene of interest, which you select for in progenies. And you have a new plant variety. And you've accomplished what you set out to do, but you've accomplished a lot of other things as well. In other words, you've transferred a lot of other DNA to the product that you didn't intend to. Now, some of that can be solved by inbreeding. So you can take this recombinant plant, this F1, and you can inbreed it for as many generations as you like. And the more you inbreed, the more homologous the chromosome will become. You'll end up having an essentially all green chromosome, but you can never get it completely homologous by inbreeding. And so you'll always have unknown genetic changes that you can't control. Now, using plant biotechnology, you can do the same thing, but with much greater precision. So again, if you start out with a plant with a yellow chromosome and your desired gene, the orange dot, you clone it. Throw away everything except the gene of interest. Then you can transfer that cloned gene specifically into the plant with the green chromosome, so that you end up with a single gene transfer, and you know that the only change you've made is the addition of that one gene. There are no off-target effects from your genetic engineering. Now, on the right here, I simply wanted to make the point that using conventional breeding, I mean, people always worry about in breeding plants, if you're making something toxic. And it's just as easy to make something toxic by conventional breeding as it is with recombinant DNA breeding. In fact, probably even more so. And a good example is potatoes. Potatoes produce alkaloids, like the structure you see here, which is poisonous. And it's poisonous to insects, so potatoes have been bred for high glycoalkaloid content so that they're resistant to insects. But this bar graph at the bottom here shows that disease resistance or pest resistance is correlated with toxicity to humans. So potatoes that were bred for high glycoalkaloids were never put on the market because they're too toxic to eat. And that was done entirely by traditional sperm on egg methods. No recombinant DNA at all. So my point is, it's equally possible to make something dangerous with either technology. Okay, what's the definition of a GMO? Any organism whose genetic material has been altered using genetic engineering techniques. Does not occur naturally by mating or natural recombination. On the left, you see the steps for producing a recombinant plant. You have an agricultural need. You isolate a gene that controls the trait of interest. You insert that gene into a cloning vector. Then there are a number of ways that you can get that cloned vector into a plant. There's such a thing as a gene gun that can shoot the DNA in. More commonly now, it's put into the plasmid is put into a bacterial cell, which then transfers it to a plant cell. Then after the GMO is produced, it's tested for efficacy. Does it solve the problem? And for safety. And it has to be approved in the US by these government agencies: the Environmental Protection Agency, the Food and Drug Administration, and the US Department of Agriculture. So all of these government agencies have to evaluate the data behind a particular GMO and approve it before it can be produced commercially. The important point to make here is that this applies to only recombinant GMOs. Plant products made by traditional methods do not have to have approval from all of these agencies. So where do we stand in terms of crops in the US? The main ones that are genetically modified are corn, soybean, and cotton. These, the modifications are primarily for pest resistance. And if you look at the graph on the right, you can see that over time, this stops in 2016, it's even further now, but it's plateauing toward 100%. So well over 90% of all soybeans, all cotton, and all corn contain recombinant DNA molecules. So you really can't exist without encountering GMOs. And if you just look at food labels, when you go to the store, many of them will say non-GMO, which implies that if it was a GMO, it would be dangerous, which is not true. There is no evidence after over 30 years of growing GMOs and eating GMOs that they are toxic or in any way bad for health of either plants or of either humans or domestic animals. So why do farmers choose GMO crops? Well, to manage weeds, to control insects, prevent diseases, and increase productivity or enhance nutritional content. So the first three of these are of benefit to farmers, but to improve nutritional content, it's not necessarily a benefit to farmers, but it could be a big benefit to consumers. So what are the advantages of genetic engineering? Just to make 2 points clear. First of all, genetic engineering is much faster. The engineering is done in a matter of months or at most a couple of years, as opposed to years or decades to transfer a trait by traditional breeding. And the second point I made earlier, it's gene specific. So with genetic engineering, you know the gene that you're transferring. You know that's the only thing you transferred. So there are no off-target genes in the product. Now, to give you an example, genetic engineering of golden rice to produce vitamin A. What is golden rice? Well, you see in the upper right here, some white rice and some yellow rice. The yellow rice is golden. And why is it yellow? Because vitamin A is yellow, and this rice overproduces vitamin A. Now, if you go to the left-hand side, vitamin A deficiency kills over 1 million people a year and causes blindness in roughly 500,000 children per year, and it occurs in approximately 120 countries. And you see in this map here where vitamin A deficiency is prevalent around the world. Now, rice provides 30 to 70% of the energy intake for people in Asian countries. So what has been done? Well, starting in about the year 2000, these two people, Ingo Potrykus and Peter Bayer, started trying to engineer rice, genetically engineer rice, to overproduce vitamin A. And on the lower right here, you can see how they did it. This is the pathway for vitamin A biosynthesis. And rice doesn't have it. It's missing a couple of enzymes. So what Potrikis and Bayer did was take a couple of cloned genes, one from a flower, a daffodil, called, it encodes an enzyme called phytoene synthase, and another gene from a bacterium, Erwinia, called phytoene desaturase, and genetically engineered these two genes into the genome of rice so that now rice can make vitamin A. And interestingly, they put these two genes under the control of endosperm-specific promoters. Now, endosperm is found only in seeds, and an endosperm-specific promoter is on, in other words, the gene is operating only in the endosperm, not in any other part of the plant, not in roots, not leaves, not stems, not flowers, only in the endosperm of seeds. So, but because of that overproduction in seeds, the seeds turn yellow because they have vitamin A, a lot of vitamin A.
Professor?
Yes?
So that would mean that the golden rice is considered a GMO?
Yes. Yeah, that's the essential point here. Golden rice is a GMO. Could not have been produced in any other way. You couldn't do this by traditional breeding. And the reason these two people did it is because they recognized the need worldwide for the solution to vitamin A deficiency. So if you go over to the left again, golden rice provides the entire recommended daily allowance of vitamin A at a consumption of about a cup per day. The seeds for it are distributed to free of charge to subsistence level farmers. They really aren't, but it's been proposed that they should be. No company would receive royalties. The humanitarian versus commercial cutoff set at about $10,000 per farmer. So why isn't a golden rice produced throughout the portions of the world that could use it? Because it's controversial, because of the point you just made, it's a GMO. And the reason that it's not being produced, even though it's been available for many years, is because some organizations like Greenpeace and Friends of the Earth, who are opposed to GMO technology in any form, they have lobbied governments throughout the developing world to not allow golden rice production because it is a GMO. And the main reason they're opposed to it is that even though they acknowledge it would do a lot of good, they're afraid it would be a slippery slope. It would open the door to more widespread use of GMOs. And this is despite the fact that I showed you on a previous slide. Here, despite the fact that some of the main crops throughout the world are already almost entirely GMOs. And many of these are eaten by humans with no obvious deleterious effects. So some organizations are opposed to the use of the technology, and for that reason, only two countries, the Philippines and Bangladesh, have regulatory approval pending. And I just heard recently that Bangladesh has approved the growing of golden rice in Bangladesh. But that's far from meeting the need. So the point I want to make here is that the technology is sort of beside the point. The point is, what's the product? And in red here in the center, you must weigh the benefits of any product versus its liabilities or potential liabilities, and then decide, do the benefits outweigh the liabilities? On this slide, what are the pros? Why do people advocate the use of GMOs, and what are the cons? Well, as I've already said, you can make crops that are resistant to various different kinds of pests, improve nutritions. I've just given you an example of improved vitamin content. And as I said earlier, there has been no case of toxicity in food that I am aware of, even though we've been eating these things for decades. And so why are people opposed? There is a good argument that the pests that the crop is resistant to, the pest could become resistant. And that does happen. It has happened, but there are ways around it. I won't go into those, but if pesticide, if pests develop resistance, there are ways to deal with that. There's a legitimate concern about with crop plants about the development of so-called super weeds, that a gene in a GMO, a recombinant gene, could escape through pollen to weeds in the neighborhood and make them into super weeds. To my knowledge, even though that there's been a lot of field work done looking for these things, it hasn't happened. Some people argue that there are alternative approaches to food production that would be better. And if there are, we would like to see them. And some people are simply mistrustful of the data themselves, the data that are published in peer-reviewed journals. And some people have an inherent distrust of agribusiness, and it's true that it's only agribusiness that can afford to develop GMOs and market them. But again, I want to come back to the conclusion that I stated earlier, that this controversy, pros and cons, it just misses the point. The point is the product, not the technology. So each new product should be evaluated on its merits, not by the technology used to produce it. If the benefits outweigh the liabilities, then a regulatory agency might give approval. If the liabilities seem too great, then no, they wouldn't. But again, humans have to make these decisions. The technology stands alone, and we can use it for whatever purpose we wish, and we just want to make sure that nothing dangerous is ever done. Okay, I have a summary slide here that you can review. Just reiterates the points, but this is a study aid for you. And now I want to stop and ask, are there any questions about any of these slides on molecular genetics? Okay, if not, I would like to stop sharing this screen. And go back to Mendelian genetics, because Scott, are you here?
Yes, sir.
Okay. Scott has dealt in his workshops with the analysis of segregating progenies, and he'd like to go over some of these slides that I sort of glossed over before. So I'm going to share my screen again. and call up Mendelian inheritance. And first, make a point about Mendel's laws of inheritance. You need to know this. The laws involve segregation. Segregation occurs at telophase 2 in meiosis. Dominance, a dominant allele completely masks the effect of a recessive one. And independent assortment of chromosomes, alleles on different chromosomes, that occurs in metaphase 1 of meiosis. And there is a summary slide which you can go over on your own time. But then we have some examples that I didn't really go over. And Scott, are you prepared to discuss some of these?
I think I can, yes.
Okay, if you need a slide, I can go either forward or backward, or maybe you can do it if you have control of the slides.
You would have to give me screen sharing permission because these are two Punnett squares and they're probably easier to.
Okay, you did this last time using, I think, a whiteboard, right?
Yes, sir.
Can you do that again?
Well, let's see. I'm not able to access a whiteboard.
Okay, well, okay, then let me stop sharing this screen and share the worksheet. You should be able to see the worksheet of Mendelian genetics. Can you see that?
Yes, sir.
Okay, so in red here, I've given the answers to the questions I asked on that. And I made a little drawing here. I think, Scott, that's what you had in mind doing, right?
Yes, sir. I was going to lay out a Punnett square.
Okay, this is a Punnett square. It's labeled sperm on one side, and the other side should say egg. I didn't put that in. But this is an answer to a question about breeding. So, Scott, have you reviewed this? Are you familiar with it, or would you like me to describe it?
No, this seems to be a pretty basic monohybrid problem. Are we talking about the tall plants? Yeah.
Yeah, tall and short plants.
Okay. So, yeah, you're doing Mendelian genetics, you have to remember we have pairs of chromosomes. These organisms have pairs of chromosomes. And who remembers what homozygous meant?
It contained the same gene, like capital T, capital T.
Yeah, it can, and so let's, the gene is for height, for example, but probably a better term would be allele, where we're talking about an allele. Does anybody remember the definition of an allele?
I think so. It's P, capital P, capital, and P, capital, and P small, right? And then we do like a cross, whatever.
We can, yeah, we're going to get to that, but I'm just looking right now. Does anybody remember the definition for an allele?
Isn't it just one of the genes like inherited from one of the parents or both?
It can be, yeah. It specifically refers to variations on a gene. So you might have tall and short are variations on this plant height gene. So tall would be one allele and short would be another allele. But it's still contained within the same locus or the gene for plant height. So in this case, we're looking just at plant height, so we'll assume the alleles are only for height, and it looks like we have tall being a dominant trait, and so it was assigned to the capital T. When you talk about traits, we have dominant and recessive traits, or dominant and recessive alleles is probably a better way to say it. By convention, when we talk about a dominant allele, we usually assign it a capital letter. And when we talk about a recessive allele, we usually give it a lowercase letter. Now, these letters should be the same because we are simply talking about this different variants of the same gene. So you would not, if you're looking at this, you would not say capital T is tall and small s is short, because it's still we're still referring to height. So we would use a capital T for tall and a lowercase t for short. Or whatever you choose, it's just the letter should be the same and make sense to you. Are we following along with this so far?
Yeah, it's clear.
Okay. So we see in the problem it says the tall is the dominant trait, and they arbitrarily assigned it the genotype of capital T. The alternate trait is short, therefore it is recessive, and it was given the lowercase t. So if we have, where is the, I am having a difficulty reading this.
I don't know if I can make it bigger.
You
This-
can borrow
okay.
my you can borrow my eyeglasses.
I've got eyeglasses. That's the problem. I need a magnifying glass. So what it's saying in the setup is that the tall pea plant was inbred for 6 generations. In other words, after 6 generations, all this ever produced was tall plants. And so therefore it's inbred, and we can assume that it would continue to produce tall plants. And what we don't know is what its actual genotype is. A corresponding short plant is also bred for 6 generations, and it continues to make short plants. Now, in order for this to produce short plants, if we're assuming what they call complete dominance, complete dominance it's a safe thing to assume unless you are told something different in the problem setup. But complete dominance means that if you have one dominant copy of the two alleles, you will display the dominant trait. And in this case, tall appears to be the dominant. Well, we don't know. Tall is a trait. What that means is, what would be the term for a plant that displayed two of the same alleles for tall?
Just homozygous.
Okay, homozygous. Generally, when we talk about homozygous, we're talking about either dominant or recessive, because we have to specify. Saying homozygous just means I have two of the same things, so therefore I have to specify. Am I talking two of the same things with a big T in this case, or two of the same things with a little t? So we use recessive and dominant as well. So I'll try and avoid talking about homozygous as a single term. Which term, though, can we use as just a single term? It needs nothing else. We have homozygous and we have heterozygous. So what does heterozygous mean?
It's 50-50, right? It's small and tall.
Exactly. It's got one of each allele. And so when we say heterozygous, that's all this, we're being specific. It means it has one of each allele. So we don't have to specify whether we're talking dominant or recessive. But with homozygous, we need to say that. All right. So we are going to look at crossing these two plants and see what we get with the F1s. What was F1? What did that mean?
The generation.
Yeah, it's the generation. So, and the number indicates which generation it is after the cross. So if we cross two parents, the offspring would be known as F1s, filial generation 1. And if we cross the F1s, we would get filial generation 2. So the number simply indicates how far down the rabbit hole we've taken these crossings. So we need to lay out this problem, and we're going to have to set up our genotypes based on the information given. We have tall as the dominant trait. So what symbol did we give it?
Could you repeat what you said?
Sure. Tall, we said tall was the dominant trait. And so what genotype are we going to assign to the plant that has, for 6 generations, produced nothing but tall?
2 capital T's.
Okay, 2 capital T's. And what genotype are we going to assign to the plant that has produced, for 6 generations, nothing but short plants?
2 lowercase t's.
2 lowercase t's. That's excellent. Now, if those two things are crossed, do you know how to set out the Punnett squares? Have you played with that before?
Yeah. One of the parents' alleles go on the top and the other one goes on the side.
Excellent. And then you simply look at the box. You bring over the letter from the left and the letter from the top. You bring it down into that first empty box, and then you come out with, say, on one side, on the left side, we might have T, and on the right side, or the top side, we might have lowercase t and lowercase t. So what will every one of the F1 generations have? If one parent is contributing nothing but capital F's, and one parent is contributing T's, and one parent is contributing nothing but lowercase t's. This is where the whiteboard comes in handy, but we don't have that.
Would you get two dominant sets of F2s with like capital T's and then the second one with lowercase t's?
So we're talking about F1s, but no, what we were going to end up with is a bunch of 100% what they call heterozygous. They're going to each have a capital F, or I keep saying F, capital T and a lowercase t. So these are all heterozygotes. And that is what you typically see, or you will see, whenever you take a true breeding dominant and you put it with a true breeding recessive, your F1 generation will be nothing but 100% heterozygotes. It's not until the F2 generation that we start to see things break out a little bit. And that's actually what this Punnett square is down at the bottom, where we're taking, if you look at where it says sperm on the top, we have a capital T and a lowercase t. So what kind of organism has a capital and a lowercase? What's the name for that kind of genetic display?
Phenotype, right?
Phenotype would be what we would see, where you would use words to describe what you see. But we're looking at, let's go back to heterozygous or
Oh, okay.
homozygous dominant or homozygous recessive. So what organisms, so I wasn't clear enough, but what organism, what would you call an organism that displayed a genotype of a capital T and a lowercase t?
Heterozygous.
Yes, heterozygous. So on the top, I have one heterozygous individual, and on the left side, I have the same. The second individual is also heterozygous. That makes sense because we're crossing two of our F1 generations, and remember, they were 100% heterozygous. If I cross, I go, say I go from the capital T on the top of the left, and I move over to the first, I could put in that capital T, I basically transfer it over, and then I take the second letter, because remember, we have to have pairs of chromosomes, one from each parent. I'm going to take the capital T and I'm going to bring that capital T from the first column down, giving me capital T in red. Are you following that?
Yeah.
And that individual is known as what? Capital T, capital T?
Homozygous dominant.
Homozygous dominant, okay. What about this next one? We carry the capital T over again, one more column, and then the one above it is a lowercase t. We have a capital T, lowercase t, so that is another heterozygote. We go to the next row down, where on the left, which is the lowercase t, we transfer a lowercase t over. A capital T comes down from the top. We have another heterozygous individual. And then in the last box, we would fill in with a lowercase t, a lowercase t. So that's how a Punnett square works. And this monohybrid crosses, they're fairly easy to deal with because you're only looking at one trait at a time. But what we would notice then is a common question is, what is the genotype ratio or genotypic ratio for this F2 generation? And that's the answer that you're going to get out of the red in that box, in the Punnett square we just filled out. So what is the genotypic ratio of the F2s?
Is it just 3 to 1?
Anybody else? Anybody agree?
Yeah,
Anybody have
75 to 25%. It's
Okay.
3 to 1, I think so, yeah.
All right, so let's listen careful to the words. I asked what the genotypic ratio would be, not the phenotypic ratio. So these are where the questions, you got to read them real careful or you get tripped up. What is genotype asking for?
The alleles that the organisms have.
Correct. So how many alleles does the organism have? How many total combinations does it have here?
3.
3. So the first one would be 1. This is a what? Homozygous dominant. Then we would list how many heterozygotes, and then we would list how many homozygous recessives we have. So in this case, we have 1 homozygous dominant. So we'd have 1 with a colon. This is a ratio, so 1 to 2. Here is a heterozygote, and here is a heterozygote. So 1 to 2, 2, we have 1 homozygous recessive. 1 to 2 to 1. That would be how you would answer a genotypic ratio for the F2s.
You say 1 to 2 to 1?
Yes.
Cool.
Now you could be more specific by saying 1 homozygous dominant, 2 heterozygotes, 1 homozygous recessive. But it is pretty much standard. If you see 1 to 2 to 1, it's pretty much understood that you're talking about dominants, homozygous dominants, homozygous or heterozygotes and homozygous recessives in that order. So this is how you would lay out the Punnett square and answer a question on genotype. But they could also ask you, what is the genotypic ratio? Or the phenotypic ratio. What is the phenotypic ratio?
The phenotypic ratio would actually be 3 to 1, right?
Yes, 3 to 1, and I would say 3 purple to 1, or 3 tall to 1 short. I'm going back and forth between 2 problems here. So I would say your phenotype would be 3 tall, colon, 1 short. All right, that assumes complete dominance. And what did we say complete dominance was? Having one of the dominant alleles means what? That we're going to display the dominant phenotype. As long as I have one dominant allele, I will display a dominant phenotype in complete dominance. And there's different types of dominance, and those are important to know as well. And that was actually in some of your slides, and we can, I guess we're out of time to cover it. If you have incomplete dominance, you end up with a blending. Say you had something that was red as a dominant color and blue as a recessive color. The intermediate blended form would be purple in incomplete dominance, because neither one wins. They both contribute equally. There is codominance, in which essentially they both win. And if you had that same kind of flower, for instance, with red and blue, you would not have purple, you would have a flower with red spots and blue spots on it. Each one of those things shows up equally because they're co-dominant. They're sharing. And you can, well, we don't even really have time to get into sex-linked or the polygenetic inheritance, because we're out of class time.
Yeah, we are out of class time. So thank you, Scott. Yeah, we have to crank it up now. And remember that, remember to take quiz 6 sometime this afternoon or sometime before midnight tonight.
Just before everybody goes, I can talk more about this in workshop. Because these are important topics. The types of dominance, they're not difficult, but I will cover them in my workshop at 2. So if you have a chance, please attend.
Okay. Anything else before I'm going to stop sharing and stop recording?